Hey everyone, I just wanted to hop on and talk a little bit about DNA today. So DNA does a bunch of things for our body, but particularly I'm going to talk about replication, transcription, and translation of DNA. So replication. The whole purpose of our DNA is that it grows, it builds our body, it builds our cells. So our DNA obviously has to reproduce. It goes through a process called replication. The DNA basically is split in half, and then new nucleotides are attached on to form new DNA. So then we have transcription. So transcription is when you take that half DNA where it's uncoiled, ripped apart, and then you make RNA. So the RNA that we're going to talk about in particular is messenger RNA. You have a strand of RNA that will travel out into the cell. Once it gets to its location, then you have to make proteins. Each set of 3 nucleotides makes a codon, and the codon codes for an amino acid. And when you put together a bunch of amino acids, you get a protein. So there are 4 codons that you really need to know. One codon is actually going to start the process of translation, and there's 3 different codons that can stop the process altogether. So the codon that will start a translation is AUG, like August, AUG. So this will start the translation. And the translation will go down the string, adding amino acids to each triplet of nucleotides, or a codon, until it reaches these stop codons. Now there's 3 stop codons, like I said. There's UAG, UGA, and UAA. When it reaches these codons, the process is stopped and the protein is done. So the biggest thing here is we have a code, and then it reads that code, attaches these amino acids, and causes a large protein. And this process is called translation. And then this is where we run into the problem of deletion and insertion, is with the stop codon and the start codon. Because if you had a protein that was supposed to be made out of 20 amino acids, but then you had an insertion of 1 nucleotide somewhere near the beginning of this chain, you could have a shift of these triplets where you could actually get a different code halfway through the segment that will stop the process of making these nucleotide chains.